Well, hi everyone. Welcome to another edition of the GNE Symposium Speaker Series. I'm filling in for Noah Wise later today. And our guest speaker today is Rebecca Schmitt from University of Indiana, who works in the Doles lab. Just a quick background. Rebecca received a PhD in human genetics from Virginia Commonwealth University, investigating genetics and environmental factors that influence Drosophila ethanol sedation. She started her postdoc position with Jason Doles at the Mayo Clinic, where she began looking into hereditary myopathies, and I believe she moved with Jason to Indiana. Is that right?
Yep, that's correct. We moved here just over a year ago, almost a
Oh,
year and a half.
really?
Yep.
Oh, cool. That's a big change.
It has been a big change, but it's always nice to kind of mix things up a little bit here and there. So it's been good.
Oh, great. Good to hear.
All
Okay.
right.
And how many years of postdoc have you done now?
So I have been a postdoc for about 4.5 years now.
Cool.
Um, yeah, so this is my fifth year. I always have to count. uh, yeah, and it's all been in Jason's lab. I haven't done a postdoc somewhere else yet, so still in the same place, but I like it
Gotcha.
here. Oh, oh, sorry. Okay, I can share my screen. All right. Oh, I know how to do this. All right. Um, all right. I guess I will get started. If anyone has any questions, I will try to monitor for the Q&A. So feel free to ask questions at any time, or feel free to jump in if you can. But thank you for the introduction. As we were introduced, my name is Becky. And so today I'm going to be telling you guys a little bit about my project that is exploring genetic changes in cell recycling in GNE myopathy. I have it geared a little bit more towards non-science folks, but please, like I said, feel free to ask any questions. So just as a brief overview of the topics that I'm going to go into covering today is just a brief introduction of GNE myopathy, go into a little bit of information about some of the research challenges and opportunities in the field, as well as talk about the model that we're using and the experimental plan that we've been using to study GNE myopathy. And then specifically going into some of the mechanisms that we're interested in, such as autophagy, and I'll explain more about what that is, as well as going on to then some conclusions and future directions. All right, so starting with the two beginning pieces, I didn't want to go too much into the GNE myopathy background, because I'm sure many people are familiar with this, but GNE is a gene that encodes the UDP and acetylglucosamine 2-epimerase and acetylminosamine kinase gene. As an enzyme, it is part of the sialic acid biosynthesis. And so sialic acid is an important sugar that is on the terminal end of many lipids and proteins that are involved in many different biological processes that are involved for signaling and so forth. Relatively, GNE myopathy is rare. It's considered to affect about 5 people per 1 million. There is a difference when we start looking at different ethnic populations. There is higher prevalence in certain populations compared to others, but overall on a global level, it is a relatively rare myopathy. The presentation of GNE myopathy typically starts, or the standard presentation is distal muscle weakness or foot drop. Over time, this muscle weakness does spread up the legs as well as can affect the upper body over time. And then for the diagnosis, there are 3 really main key pieces that are looked for. So one of the first is the genetics. So GNE myopathy is a recessive condition, meaning that both the alleles a person carries needs to both contain at least 1 mutation. And so typically sequencing is done to find these or identify these mutations. And so far to date, there's been more than 200 mutations found across the GNE gene. In addition to this, there are the clinical features, as I had previously mentioned. Specifically, foot drop is one of the main beginning characteristics. And then there is also muscle histopathology that can be assessed. So there is myofiber atrophy, which includes the muscle getting smaller. There's different molecular markers, and then rimmed vacuoles are one of the main hallmarks. And so in this image here on the right, these are two GNE myopathy patient samples that we have through our collaborator. And the arrows are pointing to these said rim vacuoles. And so really this is just protein aggregates that are bound together. And then it's believed that during the actual processing for the hematoxylin and eosin staining that is shown here, that the center is actually taken out or through the staining process. And so that's why we see kind of these red granules on the outside with this empty center. So there are some significant challenges in the GNE myopathy research field. And so one of those, I believe, is the mouse model. So there are some mouse models out there available for studying GNE myopathy. They have, in my mind, some significant limitations, such as developing kidney disease, which is something that is not standardly seen in the patient population, as well as the cost of some of the models that do develop myopathy. It just makes it a more challenge to do in a research setting. In addition, as I mentioned, the patient population is relatively rare, so the samples are rare. So we can't just get an unlimited source of samples. And in addition to this, the clinical trials that have been undergone, they do show some promising results, but also at the same time, maybe not necessarily something, or I guess could be something that we could work a little bit more towards finding additional pathways to improve patient outcome. So because of these challenges, that leads us to different sets of opportunities. So one of the first ones is cell-based modeling. And so I'll be talking about this quite a bit, as this is what my project is revolving around. And then in addition, this allows for a more substantial area in the therapeutics, is identifying the mechanisms and attempting to find different therapeutic options for patients. So next, I'm going to tell you guys a little bit about the model that we're using. And so this model is a cell-based model. And so before going into explaining why we're using the cell-based model, I wanted to explain a little bit about what it is. So we use specifically induced pluripotent stem cells. And so I want to start with explaining what pluripotent stem cells are. So stem cells have two main characteristics, and one of the first of those is that they have the ability to self-renew. So they're able to essentially continue their cell population so that we can have an unlimited source of cells. And then the second part is that these pluripotent stem cells are able to differentiate. And so that's where the pluripotency comes in, because they're able to differentiate into these more mature cells like neurons or muscle cells and so forth. The induced part of induced pluripotent stem cells is describing the fact that we are taking these cells from an adult population or adult cells. And so we specifically in our studies have used skin biopsies that were taken, and then the skin cells isolated. And then they undergo what is called reprogramming, which essentially takes the skin cells that are in a mature cell state, and it kind of reverts them back into being more immature. And so that's where we get the induced pluripotent stem cells from. So the reasons why we use this model, some of them I've alluded to, but is the fact that we can take these iPSCs, which are more immature in nature, and differentiate them into a bunch of cell types that are more mature. So specifically in my studies, we'll be looking at muscle cells. In addition, there is basically an unlimited supply of these. Because of their ability to self-renew, we can constantly have samples that will not run out. In addition, we are using cells from human, which is a benefit from using cells from some sort of other model or other, I guess, organisms such as mice. In addition, that these are also taken from adult cells. And this model can also be more cost effective compared to other model systems. And so for us, this really, and I guess many other fields, this isn't a novel approach by any means. Many people do this, but it's something that we have now applied to the GNE myopathy field. So I wanted to talk a little bit about the experimental plan and why muscle is important. So muscle is important for many activities. So typically we think of movement, such as running, biking, even breathing. So the diaphragm is a muscle and is necessary for our ability to breathe. In addition, daily care. So anything from getting in and out of bed, to showering, to brushing your teeth, all of these daily activities or self-care activities typically involve some sort of muscle movement. And then there are activities that say people like, such as employment, which is also very important. Anything from things you enjoy or a person may enjoy, such as dancing or hiking. So muscle is an aspect of everyday life for the vast majority of people. And so anything that affects muscle is can typically be somewhat of an issue. So what we are doing is we are studying skeletal muscle. And so instead of using muscle from patients, we're able to model muscle in a dish. And so the way we do this is we take the immature induced in pluripotent stem cells, and we're able to treat these stem cells with a bunch of media cocktails. And this allows them to undergo a differentiation process into cells that are called myotubes. And so myotubes aren't exactly like muscle, but they are similar in the fact that they're multinucleated, meaning they have more than one nucleus, and essentially is the best way that we are able to right now model muscle in a dish. And so what we can ask then is when we take these induced pluripotent stem cells, we can ask if mutations or changes that are in the GNE gene impacts the ability of muscle cell formation. And so we've been studying particularly this end stage, mature muscle cell, which I will refer to as myotubes. But I wanted to just share briefly one of the ways that we are able to study this, and this is through a method that allows us to visualize these cells called immunofluorescence. And so essentially what that allows us to do is use a marker, which is called myosin heavy chain. It stands for a protein that's involved in muscle function. And essentially we use this just to demonstrate that the cells we are looking at are mature. And so I will refer to these more mature muscle cells as myotubes or expressing myosin heavy chain going forward. So one of the first things we want to know is, in our studies, are we able to actually take induced pluripotent stem cells and do this differentiation down to these muscle, more mature muscle cells or myotubes? So to do this, we first use some control samples. And so these are from different patients that do not have GNE myopathy. And so they do not have mutations in GNE. And so what we were able to find was that we were in fact able to differentiate these iPSCs into muscle cells, which is depicted here in these two images where we can see these tubular-like structures that are stained in green, which is the myosin heavy chain, indicating mature muscle cells have been formed. On these images is also something called DAPI. And so that is just staining for the nuclei of the cells. So next, going into the patient samples that we do have, because we do want to study the GNE mutations, we were able to get some patient samples. And I wanted to tell you guys a little bit about these patients. But first, I wanted to explain a little bit about the GNE protein. So the GNE protein is actually a bifunctional enzyme, which means that it is one enzyme, but it has two functions. So the first part of the gene is an epimerase. And so essentially that just means it has its own function. Well, the second half of the enzyme is a kinase. And so that has its own function. So when we're considering the mutations that patients can have, because it is recessive, that means both alleles that they have to have mutations. And that allows us to see a different variety of patient mutations, such that they have two of the same mutations, they have different mutations in the same domain, or different mutations in either the epimerase and kinase domain. So it's kind of a wide variety that can occur here. But so our patient 1, which I will be referred to as GNE1, is a GNE myopathy patient that has one mutation in the epimerase domain, as well as one mutation in the kinase domain. The second patient sample that we have has two different mutations, but they both lie in the kinase domain. And so what we want to know is whether these mutations are able to influence the ability, iPSC ability to form muscle cells. And so that's exactly what we looked for. So this is what the images that I had shown previously with our two control lines with which do not have GNE mutations. And they were able to form these myotubes as stated previously. But when we did this with the two different GNE patient lines, there's a stark difference where in at least the first GNE patient line, we really don't see any expression of myosin heavy chain, suggesting that there's really no mature muscle cells that are being formed. In the second GNE patient samples, we do see that there is some formation of these mature muscle cells, but it doesn't appear to be anywhere near as much as what we see in the control cells. So this is suggesting to us that mutations in the GNE gene are causing a decreased ability for muscle cells to form. But why is this? And so that's really where our project has been moving forward, is to try to understand why there is this deficit in the ability to form muscle cells. So one of the cool things that we can do with our cell-based model system is during this differentiation of iPSCs to myotubes, we're able to take samples across this time frame. So we can look at control and the GNE myopathy samples at a bunch of different cell stages. And then we can go ahead and ask specifically what is different between these two cell types. And so when we did this, we could understand things or elucidate things that are happening in the control cells, but we can also then identify things that are happening just in the GNE myopathy cells. And so while there is some overlap, we are primarily interested in what's happening in the GNE myopathy cells that may not be happening in the control cells. And so one of these things that we did find was autophagy. And so before going into specifically what that could mean for GNE myopathy, I wanted to explain a little bit about what autophagy is and why we think that this may be playing a role. So that is where I'm going for my next piece, is to discuss autophagy and see whether it may play a role in GNE myopathy. So autophagy is another way of saying kind of like a cell recycling piece. And so in cells, when there is some sort of protein or there's some sort of organelle like mitochondria, there's something with these proteins or organelles that are maybe not functioning properly, or they just aren't needed anymore. And so there has to be a way to degrade these things or break them down. And so what happens in a cell is there's these vesicles that hold the cargo. And so this cargo can be really anything inside the cell, from the organelles I mentioned or proteins. And so when anything is encapsulated in these cargo, or these cargo are encapsulated, those are what needs to be broken down. And so what can happen is there's a second type of vesicle that is called a lysosome. And this is something that is very acidic, and it allows for the breakdown of these specific pieces that the cell wants to get rid of. And so what happens is there's a fusion between the cargo-containing vesicle and the lysosome. And so that allows for the breakdown of different proteins that are needed to be degraded. And so that leads to a healthy cell. It's able to get rid of things that it doesn't need or is inhibiting its ability to function. But on the other hand, if there is an issue with this process, that can lead to unhealthy cells and this buildup of different cargo-containing vesicles that may influence or impact the ability of the cell to function. And so this is something that we believe may be occurring in GNE myopathy. So one of the things that we can ask with our cell system is, do we see this kind of unhealthy status of autophagy in the GNE cells? And so one way we can do that is by looking specifically at the lysosome. It is known what the lysosome is and what many of the proteins make up this lysosome. So we're able to actually assess for some of those proteins. And one of the main ones is called LAMP1. And LAMP1 is the lysosomal associated membrane protein 1. And so this is a protein that is involved in the membrane structure of a lysosome. So since we know what this protein is, we can ask, okay, do we see an overabundance of LAMP1 expression in our GNE cells? Or In our case, we'd be looking for an engulfed cell type, which I will show you a little bit more of. So in these first two images are the control iPSCs that were differentiated to myotubes. And so these, although we don't see that they are myotubes in this image, we were able to identify the myotube structures previous to staining for LAMP1. So in this image, we are interested in looking at the green color. And so the green color is indicating the location of this LAMP1 protein. And so while LAMP1 is being expressed, it's more in a puncta fashion, which wouldn't suggest that it is being expressed as such as a lysosome, where we see it would form a circular pattern. But when we look at the GNE myopathy patient samples that were differentiated, we do start seeing this engulfment, where we see this LAMP1 staining pattern that is engulfing the nuclei, which is blue and called DAPI. And so what this suggests to us is that, this may be indeed the case, where we appear to have an increased expression of LAMP1 in a circular pattern, suggesting that there may be an increase of autophagy players. To further address whether this is something that we are seeing in GNE myopathy and subsequently the ability of cells to form mature muscle cells, we can ask if there's a correlation between the autophagy player accumulation and muscle cell formation. And so essentially what this graph is showing, when we're looking at the y-axis, there are points that are closer to this bottom part of the y-axis. This is more of cells that we would assume to be healthy, where they don't really have accumulation of autophagy. But as we move up the y-axis more towards the top, this measure is indicating that there is an increase in the accumulation of different autophagy players. And then we can ask about the myotube or muscle cell formation. So this time we're looking at the x-axis, and closer to this origin point here, where it's 0 is the indication that the muscle cells are not really formed and they're still mature. They're still immature. And then as we move along the x-axis to further on the right, we're seeing that the formation of more mature muscle cells. So when we asked about the correlation, what we saw was cells that had an increased accumulation of the autophagy players, they had a decreased ability to form muscle cells. So an example of this point would be here surrounded in yellow, where the point lies higher on the y-axis, suggesting accumulation of autophagy players. But when we look at the x-axis, it's more towards the left, suggesting immature muscle cell formation. And then vice versa with C. When there is a decrease in the amount of accumulation of autophagy players, there's an increase in the ability to form muscle cells. So again, this dot on the graph is showing an example of that, where when looking on the y-axis, it's relatively near the bottom, suggesting that there's little accumulation of autophagy players, but it is further on the right on the x-axis, suggesting mature muscle cell formation. And so we were able to see that this was a significant trend when we assessed the autophagy player accumulation and myotube differentiation ratios in controls and GNE myopathy cells combined. So what this essentially led us to wanting to know is whether autophagy can be altered to improve muscle cell formation. And so that's specifically what we did next. There are many different drugs that are available that can induce autophagy. And so during the stages of differentiation from iPSC to myotubes, we're able to take these drugs that induce autophagy and then ask, okay, if we induce autophagy during the differentiation, do we in fact see this increase in muscle cell formation? So when we did this, we have our baseline muscle cell formation. We can see in the first GNE myopathy patient-derived samples that there's very little formation of mature myotubes. There's this one myotube that is seen here. And then in the second GNE myopathy patient-derived samples, there is more myotube formation, but it's still not as much as what we would normally see in a control. So that's when we went in and we gave these cells an autophagy activator. And that's exactly what we were able to see, was that with this drug, there was an increase in myotube formation. So you can see that in both the first patient sample, we're seeing an increase in these green structures, as well as in the second myopathy, GNE myopathy patient samples. There is, again, this increase in myotube formation. So that's something that is pretty exciting, which this data would suggest that activation of autophagy in GNE myopathy can improve the myogenic outcome of these cells. So that leads us really to some of the conclusions and future directions that we have. So the overall hypotheses that we're kind of working with right now is that in people that have no mutations in GNE myopathy, essentially they're able to have this normal autophagy or normal recycling program occur in the muscle. And so that leads to healthy muscle. But when there is mutations in GNE, this appears to be causing some sort of dysfunction in autophagy, where we're not seeing that full recycling process occur, which is potentially implicated as being causative, or even just not necessarily causative, but influencing the affected muscle phenotype. And so our idea, or our hope, would be that we could target this autophagy dysfunction and maybe not fully reverse it, but hopefully enhance it to where we can see maybe a little bit more breakdown of these cargo that really need to be broken down to cause healthy cells, so that we can hopefully see improved muscle function. So our future directions really include a lot of how is GNE influencing autophagy. So I mentioned a little bit in the background, GNE is primarily known, or its main function is known, in sialic acid biosynthesis, but you know, is that really it? And so that's not something that I think is fully known. And so that is something we want to address, as well as what do the specific domains of the GNE enzyme have. So this relates to that first kind of question. You know, we know sialic acid biosynthesis, but as an epimerase and a kinase functioning protein, there could be other things that it's doing. And so can we identify specifically if one or even both of these enzymatic domains are doing something completely unrelated to sialic acid that we just have to elucidate now? And then the third kind of piece that we're interested in is looking at the mutation location. So I briefly talked a little bit about how there can be a wide variety of mutations that land in either of the domains or in the same domain. And so really, does the mutation location influence phenotype? Can this explain some of the more severe patient phenotypes compared to patients that are still affected, but maybe not as severely as others? And so really what I want to leave with, the key takeaways is that to date, so far, there's really no cures available for GNE myopathy, but we are hoping with utilization of this cell-based model, we can understand more of the mechanisms that contribute to the disease pathology. And then ideally that potentially targeting autophagy could provide a new promising therapeutic approach. So with that, there's a lot of people that have been involved in this work between here at Indiana University, my PI, Jason Doles, and then at Mayo Clinic, specifically Dr. Margarita Maloney, and the Regenerative Medicine Corps. A lot of people have contributed to this work. So clearly there'd be no way to do this without all these people. And so I guess with that, I'll take any questions if there's any.
Thank you, Rebecca. That was a really great talk. I really enjoyed it. You had very good graphics and animation, and it's really exciting that you are uncovering a different mechanistic angle to GNE myopathy that hasn't previously been explored. So, I mean, we're also, the field is so focused on, you know, sialic acid biosynthesis and not having enough sialic acid. If there are other defects besides that, it would suggest that taking supplementation for sialic acid deficits may not address the entire cause of the disease, and that there is room for additional avenues to be explored and additional potential medications. And it's exciting that there are autophagy enhancers that are currently being tested in the clinic
Yep.
for other diseases. So we could potentially, as a field, piggyback on some of those drugs. Yeah. Does anyone have any questions? I guess I'm interested to know whether you have tried, I know you've tried cell lines with patient mutations. Have you tried like a cell line that is like engineered a null mutation that doesn't express GNE at all?
So that is not something that we have done. There have been other labs that have done it. I am personally interested in not creating a null mutation for the fact that patients typically don't have null mutations. And then also when we do look in mice models, people have tried to create this null mutation and it's embryonic lethal, which would suggest that the null mutation is not compatible with life. So for me personally,
good to calibrate your assay to show that if there are cell lines that are GNE null. I believe Catherine Koswara at Yale has created some. It would be good if you're interested to just use as a calibration for your assay to show, it's like on the extreme, on your graph, like, up in the compared
no,
to
that's fair.
healthy.
That's a very good point. And that's actually a very good idea.
Yeah. So it would, the patient cells would be somewhere in the middle of healthy and the null. I think it would be nice to show that, even though I know it's not realistic and conducive to life.
But that's still a very good idea. No, I like that. That is definitely something.
Yeah, I can connect you with her. Definitely.
Yeah, that'd be awesome.
I think, there are a lot of cell lines and resources that are available in the community, and NDF would like to encourage the sharing of resources, whether they're animals or patient cells or to help across many other projects. That sounds good, and that's very exciting stuff. I mean, to uncover like a new mechanism.
That's what we're hoping for, is to help all come together and all target it from different angles. And
what about in the mouse model, tissues from mouse model? Are you planning to check any of that?
So there's been some preliminary data or some studies that have shown that there is presence of these vesicles that are seen that are thought to be related to autophagy. But there hasn't been really too much done previously, but that is something we'd like to do. We do have one of the mouse models that is commercially available from JAX, a mouse repository. So we do have that mouse model. We just have not gotten there yet.
Gotcha.
But it is something we like to do. But we did take it from us. We had just recently got it when we went to Mayo and then moving it here. So we're trying to up our colony with that before really diving in.
Gotcha. Okay.
It would be really cool to have
that. Yep. I believe Catherine is also working on tissue from the one from JAX that has the M743T Middle Eastern mutation. And some are having, just the disease, and then some treated with like GNE AAV gene therapy. So that is also available. I mean, for
that's
analysis,
awesome.
If
that
you would like to.
Really awesome.
Yes. So, yep.
Yeah, thank you. I appreciate that. I'd love to get in contact with her and see what we can do to collaborate.
Yep. Awesome. Okay. Is there any other questions from Taylor Hines? I have mutations on both the epimerase and kinase sections of the gene and seem to be on more of the severe side in terms of progression. Do we know if this is generally true?
So that is a very good question. It's something that we have looked into quite a bit, and the literature that we've seen is most suggest that they could be a little bit more severe. There are some papers that have suggested mutations in the kinase domain, either homozygous or two different mutations in the kinase domain are more severe. I would say at this point, we're not really sure, but that's something that we are looking into. So I didn't mention it a whole lot here, but that's a big part of the project that I am looking to do next, is using CRISPR-Cas9 to introduce mutations and go through systematically and assess, do mutations in one in each domain or in just one domain versus the other cause a more severe phenotype. And so I would say it's not really sure yet, but it's something that we are hopefully actually actively working on to find out.
Great. Taylor has another question. Is it your hypothesis that there is simply more cellular junk to clear out in GNE cells, or that the disease causes a failure to properly clean out cells? I think it's the latter.
Yes, that's what I would anticipate. I guess until that's fully proven, it's open to really any idea there, but that's what I would anticipate is it's failing to clear out the pieces that really need to go to keep the cell healthy. So yes, that's exactly what I would anticipate.
Cool. Great. Thank you for the questions, Taylor.
Yeah, thank you.
Okay, well, if there are no more questions, I think we can end the webinar, and I'll be getting in contact with you, Rebecca.
Thank you so much. I really appreciate it.
Okay. Yep.
Have a great rest of your day or evenings.
Yes.
Thank you so much.
Thanks, Rebecca.
Thank you.